Entitled So Professor Iradoki is currently associate professor in the School of Biomedical Engineering and Computer Engineering at Purdue University. He is also the director of the Center for Implantable Devices. He received his both of his Bachelor of Science and a Master in Electrical Engineering at the University of New Hampshire in 1997 and 1999, respectively. And he received his PhD in neuroengineering at UCLA in 2003. Currently, Dr. Iradoki's research is pursuing a set of tools to find a modular approach to the design and of biological implant for clinical treatment of psychological disorders. Today, he's going to give us a lecture about the implantable materials. Please join me in welcoming him.
Thank you very much for the kind introduction. It's very nice to be here and talk about some of the work that we're doing at Purdue in the Center for Implantable Devices and more broadly in the department. I'll begin by going through some of the work that I'm working in particularly, or that we're working in particularly in my group, but then I'll touch on some of the things that we're interested in more as a department and in directions for growth in the future. So as I know most of you are graduate students, as you're thinking about postdocs and as you're thinking about faculty positions and so on and so forth, where we have 170 faculty positions open in engineering right now. So there's opportunities. So I'll talk about that towards the end. The motivator for this is that the medical marketplace is about 19.5%, or it will be soon, about 19.5% of GDP. And that amounts to about $3 trillion a year annually. That's just in the United States. Right? And yet the way that people treat medical conditions has remained fundamentally unchanged in the last 100 years, in the sense that an individual doctor works on an individual pathology. So your cardiologist works on your heart, your neurologist works on your brain, your gastroenterologist works on your gut, and those three doctors will never talk to each other, and they don't particularly care what's going on in your heart or in your gut if they're working on your brain. These different body areas or organ areas are treated independently from one another, and the methodologies are often at odds with one another. So our sense is that we could enable multi-pathology treatment, so treatment of multiple different locations in the body, through a physically distributed network of nanoscale wireless sensors and actuators. So having a distributed network throughout your body, in your brain, your heart, your gut, and wherever, that monitors and stimulates, that senses and actuates what's going on in your body would deliver fundamentally a different way of thinking about medical treatment and eventually a paradigm shift in how medical treatment is administered and performed. And so in order to do that, we have to develop a number of different technologies. We have to develop new ways of sensing, new ways of actuating, new ways of networking devices located in disparate locations, and new ways of miniaturizing these devices so that they can go wherever you want them to go in the biology. So this is a collage of all the people that work or worked at one point. It's a little bit of a data collage now in the center, and some of the devices that we've made. But this is an enormous group effort. It's not just one person. As all of these efforts are bound to be. So I'll begin by going through some of the sensing modalities that we do, and this is really just an overview, so what I could fit into a 45 to 50-minute talk. But I'll talk about each of these different categories: sensing, actuating, networking, and miniaturization, and try to give you an overall picture of what we're trying to do. So on the sensing side, one of the projects that we're involved with is glaucoma, and in glaucoma what you want to do is you want to be able to measure the pressure inside of the eye. But the best model for doing this work in humans is actually not the rabbit, which is what most people think when they think eye surgery. They think rabbits as an animal model. It turns out the rabbit eye is quite different from the human eye. The only way in which it's similar is the size. It's about the same size, and so it's nice to work in from that perspective. But in terms of the anatomical structure, from an evolutionary perspective, we're much further from the rabbit and much closer to the mouse, which is a pain in the neck because mice are really small. So we had to go and come up with a way of measuring pressure inside of the eye of a mouse, which required us to make an entirely different, a new sensor. And so this is the sensor that we came up with. This was a collaboration between Professor Bill Chappell at Purdue. He was leading on this sensor development, and myself through a Howard Hughes funded effort in partnership with the Jackson Laboratory and Simon John out there. So this sensor is a capacitive pressure sensor. It's 500 by 500 microns. They can become as small as 300 microns, as big as 800 microns, but it gives us the sensitivity of about 0.5 millimeter of mercury that we would need when coupled with an integrated circuit to measure and log pressure changes in the eye continuously over time. So it allows us to track the results of any kind of therapy which we might want to administer for glaucoma, like drops, drug therapy, or a surgical intervention like a shunt that you might put in, or a laser trabeculectomy where you modify the biology itself to increase the amount of flow out of the eye. Another area in which we wanted to get involved with sensing, and this is actually our bread and butter, is in the neural domain. We wanted to be able to record individual unit activity in various different locations of the neural cortex. And in order to do that, you need these implanted depth electrodes. And here's a collage of various different commercially available electrodes made. University of Utah, which is now a company called BlackRock, the electrode making portion of it is. The University of Michigan made these electrodes, these and these, that are now manufactured, and these that are now manufactured by Neuronexus as a private entity as well. There's a few other designs out there from UCLA and Drexel and a couple other places. All of these electrodes have the same problem, which is that they don't work. There isn't a single commercially available neural recording electrode that works. There never has been. And that's interesting because you don't often get to make absolute blanket statements of fact in this world, right? This is one example in which you can. You can actually say all neural electrodes fail. And the reason they fail is because when you put them in the brain over a period of time, you can see that a scar forms around the area where the electrode is, and that glial scar changes the impedance and the signal-to-noise ratio of the interface between the contact point on the electrode and the cell that you're trying to record from. And it happens all of the time, right? So electrodes fail. Every now and then you see a paper that has electrodes that have been in a monkey, and they've been in a monkey for a year and they still work. The reason that happens is because you put 100 electrodes in, and you do this in 10 animals, and you'll get one animal with 2 electrodes that work in a year, right? So the yields are so catastrophically low that they're good enough to get a neat paper, but they're useless in terms of developing a device that's clinically relevant. So how do you make something clinically relevant? Well, my answer was to just avoid the problem altogether. I just thought, you know what, heck with that. I'm not going to do electrodes, because Michigan and Utah have spent tens of millions, literally tens of millions of NIH and DARPA money over the last 30 years making these electrodes, and they can't make anything that works because they have to be a certain width in order to have the rigidity to hit a deep brain structure. And once you have that width, you cause that injury, and that's it. The game is over. So I got to Purdue, and I said, I'm going to make everything that interfaces with other people's electrodes, but I'm going to make the electrode problem somebody else's problem and just steer clear, right? I'm an assistant professor. You don't want to tackle insurmountable problems. So then this guy, David Yarrow, who wasn't even in my lab, wasn't even in engineering, he's over in the ag school, he walks into my lab, and one day he says, Professor, actually walked into my office, he says, Professor, I've solved the electrode problem. And he wasn't my student. I didn't even know who he was. And my first reaction was to say, get out, I'm busy. But then you think as part of your job as a faculty member is to mentor and nurture young minds and all that good stuff. And so I thought, all right, I'm going to listen to him for 5 minutes, and then I'm going to tell him to get out. And so I said, yes, David, all right, whatever you come up with. And he said, well, the problem with these electrodes is that they're really big. So all you got to do is make them smaller. I thought, yeah. You're not the first to think of that one, right? So yeah, he said, no, but the literature shows that if something is smaller than about 20 microns in diameter, 20, 25 microns in diameter, that the brain will actually ignore it. It won't treat it as a foreign body. And so all you got to do is figure out a way to put that deep in the brain, and you're done. And I said, yeah, but that all you have to do part, that's the crux, right? Because anything, when it's 20 microns wide, I don't care how rigid or strong a material, is flexible, right? Even glass is just flexible at that thickness. So he said, well, yes, but what if instead of pushing it in the brain, we were to coat the tip with magnetic nanoparticles, and then we could use a magnetic field to pull it down into the brain? And I thought, well, that's kind of neat, right? So I said, all right, well, let's try this crazy idea of yours. And so this is in my lab. this is after a few months of development. We made a jelly mold. We got a 20,000 frame per second camera. We made a railgun. And then we tried to hit a 1 cubic millimeter target 5 centimeters down into the brain. There it is. And shot at 20,000 frames per second. There's a 20 micron electrode going exactly where we want it to go. Now, in the interest of full disclosure, I'll tell you that we shot this 12 times before we got it to go where we wanted it to go, because the aim was terrible. But this was enough to then go to DARPA and say, I need $500,000 to make this a reality, and they gave me the money because they're nice, and they're not, but they're very helpful. So they're nice in a way. And lo and behold, here we had the electrode, right? So it was a fantastic opportunity to develop the technology. We implanted it in animals. We've had them implanted in animals for 90 days at a time. And unusually, the electrodes work 100% of the time. The catch is that they're still not very accurate. And so we're working on making them more accurate. But that's another example of a sensing technology. On the actuating side, it's actually, we spend a lot more time on actuation than we do on sensing. And on sensing, we tend to partner up with folks in the biomems world to do things. But on the actuating side, the problem that we observed was a different one. So we work with a company called Cyberonics, and they funded us heavily over the last 5 years, and generously. And so the work that we've been doing with Cyberonics has been focused on trying to understand how their device works, so that we can then make it better. So what they have as a device is they have what's called a vagal nerve stimulator, which is a nerve cuff that wraps around the vagus nerve in your neck and electrically stimulates it. And that stimulus somehow goes up to the brain and does something which makes seizures happen less often. And I'm not dumbing it down from you. That's as dumb as I've got it, right? So when I first started working with them, I said, so how does your device work? No idea. So I said, all right, okay, so you're stimulating the brain. So how do you set the amplitude, right? How do you set how much current you're pushing into the body? And they said, oh, well, that's easy. We have the patient, once we've implanted them with a device, they come back in to the neurologist's office, and then we turn up the volume on the device until the patient starts to twitch, physically, visibly twitch. Then we back it off a little bit, and we leave it there. I said, okay, so what amplitude is that? No idea. Is there any change from patient to patient? Probably. I thought, okay, so, all right, so that's a little scary. And I found out later that Medtronic, with their deep brain stimulator, they do exactly the same thing. Everybody does exactly the same thing. So I said, all right, but the waveform, I mean, surely the shape of the wave, the duty cycle, the frequency, these are things that have been studied. What are they and why? And the first answer was, well, actually, we don't know what they are. We'll have to go look that up. And it took a while before we were able to dig up what the waveform actually was, which turns out not to be what it was supposed to be. However, it's been approved by the FDA. And so I asked them, I said, well, why this waveform? Why not a different waveform? And the answer was, this waveform works. If we raise the question, the FDA might come back and pull our approval and say, go figure out what's the best waveform. So we don't mess with it. These sorts of things happen a lot in the regulatory world with medical devices. And so we started to say, well, we're going to figure this out. So in addition to stimulating, so we can stimulate the vagus, we can stimulate the trigeminal, which is also useful for a bunch of different things, we would record. We would record the nerve upstream from the stimulus, or downstream, depending on which way you're interested in having the signal propagate. We would record the signal in the nerve, and then using just simple signal processing, we could break it up into different fiber types, because different fibers have different diameters. Different diameters correspond to different velocities. Different velocities means that if you record at some distance, your signals are not going to be separated in space or in time, depending on how you plot it, right? So they're going to be separated, and then we can tease them apart, with A fibers being large motor fibers, C fibers being small sensory fibers going away from the brain, and B fibers being something in the middle. And then we can start to say, well, what is the maximum amount of activation you can get at each of these fiber types? And then which fibers do you want to activate? And what % of that maximal activation do you want to achieve? And so we can determine what the stimulus is, and we can update the stimulus waveform to give you the amount of activation that we want. And if we find out that, say, 70% maximal activation of A fibers is what gives you seizure suppression, then why stimulate B and C fibers? Those go down to your gut and your heart. You don't want to be stimulating those. Actually, it's the other way around, sorry. If you want to stimulate C fibers, A and B fibers go down to your heart and your gut. They change your heart rate, they change your digestive patterns. You don't want to be activating those. Why don't we focus on the ones we want, meaning we have less current, less dosing, and a better longevity for the device, and simultaneously a better therapeutic efficacy. Moreover, when we say, all right, we're going to do 60%, say 60% A fiber activation and 45% C fiber, which works pretty well, we're now going to say that that level of therapeutic activation is what leads to the reduction in seizures. So we're not going to dial it up until you twitch. We're just going to give you 60%, and you're not going to feel it. But you don't need more, because more doesn't help you. And when we implant this thing, we're going to set it to 60%. But as you age, as you gain weight, as you dehydrate or rehydrate, as your electrode becomes encapsulated with a glial scar, it's automatically going to update the stimulus parameters, because it doesn't care what the stimulus is within safe parameters. It just wants to achieve that same amount of neural activation, which leads to the therapeutic effect. And that actually works out really well. And it doesn't just work out for epilepsy. I apologize for the print size, but there's so many things that it works out well that I wanted to stuff them all in one slide. And here they are. It looks better in a big poster. But we've been trying this out in epilepsy. We've been doing it in alcoholism, where we have alcoholic rats that we can stimulate and get them to stop drinking. We're using this in depression. We're using this in gastroparesis. So you hear a lot about diabetes, and I've avoided diabetes for years because I wanted to stay focused on the neural world. Well, it turns out one of the complications of diabetes is that the peripheral neuropathy that affects your extremities also affects your intestinal tract. And so patients that eat, the food goes into the stomach, and then it's supposed to get pushed out by the muscles into the small intestine and then down. That doesn't happen. It sits in your stomach. And then after a while, your stomach, your body does what it knows to do when things are just sitting there. It pushes it out this way. And so you go into uncontrolling bursts of vomiting that happen every 3 or 4 or 5 minutes for 3 or 4 days. And this happens once a month. 10,000 hospitalizations every year in the United States is a primary cause, and 130,000 hospitalizations every year is a secondary cause. And that's 50% more than 5 years ago. So it's a bad problem, and it's getting rapidly worse. And this helps that also. We can also add a second loop. So we have the first loop, which is to make sure that we have the same amount of excitation in the body that we want, and that we maintain that. The second loop is that we stimulate only when it's necessary. So for example, in epilepsy, we can track a third-party biomarker, like for example, seizure onset, and we can stimulate only when a seizure is about to begin. Why stimulate all the time? You're not having seizures all the time. You're having a seizure once every few days. If you're really bad, you're having 3 or 4 a day, but that's still not all the time. Right? Stimulate only when you need to, stimulate only the amount that you need to, and you minimize the dose. And that means that over time, the patient is going to become desensitized less than they would otherwise, or not at all. Another area where we're working on stimulation is a new field called optogenetics. And optogenetics is this idea that you can take the genetic code, originally from algae actually, that encodes for ion channels. So you have ion channels in your body that are voltage sensitive. You have ion channels in your body that are ligand gated or chemically sensitive. They respond to neurotransmitter concentrations, for example. There's a third kind of ion channel in algae that responds to light. And so what you can do is you can take the genetic code for that ion channel and put it in a mammalian neuron. So the neurons that get this opsin, and there's different kinds. There's excitatory ones, there's inhibitory ones, channel rhodopsin, halo rhodopsin. But the neurons that get this opsin now become light sensitive. And the reason this is interesting is because you can make a very small spot of light on a brain. So if you electrically stimulate, you electrically excite a volume of tissue around your electrode tip. If you do it with an optical fiber, it can be just a spot, just a few microns in diameter. And even better, you can separate the viral vector you use to deliver this opsin so that you have a viral vector that encodes for the enzyme that brings the opsin into the membrane, and a viral vector that encodes for the opsin itself. You can put one in over here, one in over there, and then only the neurons that go from here to here will get both viral vectors that they need, and only those neurons will become light sensitive. So when you now stimulate in the brain, you can selectively stimulate a specific tract of fibers going from one place to another in isolation of everything else around it. So it allows you to identify the circuits that you're interested in activating. This isn't something you're probably going to do in a human clinical setting, but in an animal research setting, it allows you to understand the mechanisms behind the therapy you want to deliver, so you can make your electrical implant accurately to deliver that therapy. So it helps us understand how to better design our conventional medical devices. So this is the state of the art. It's a large laser with a cable on an optical fiber and a large head mount on a rat. You can see that it's rather cumbersome. So what we wanted to do is we wanted to make something smaller. So we made something that, here it is on a quarter. It's got a microcontroller, it's wireless, wirelessly powered, and so on. And if we use a brain slice that's optogenetic and we give it blue light, every time you see a little blue dot, that's a spike of blue light, then that activates the excitatory channel rhodopsin and the neuron fires an action potential. If we give it inhibitory yellow light, then it activates the halorhodopsins, and a neuron that would ordinarily be firing becomes silent whenever we're stimulating. So it works quite well. But that's for a brain slice. What we really want to be able to do is to see if we can modify behavior in a full animal, right? A freely behaving animal. So we take these devices, we put it on a mouse, and he's running around. When the blue light goes on, we begin to stimulate, and he begins to go in left-hand circles. And whether he likes it or not, or whether he wants to or not, we can make him move in left-hand circles because we're stimulating the motor cortex, right? We did this in a couple of different animals, and it worked quite well. We moved from there to doing this in trying to modulate behavior through reward mechanisms. And so you can do conditioned place preference or conditioned place aversion, where you stimulate the reward centers of the brain to promote preference, or the fear centers of the brain to promote fear. There he goes in left-hand circles again. And you can tell he doesn't want to. So to promote fear, that's a little sadistic, but to promote fear. And so you can get a mouse that would ordinarily want to be in a dark room versus a light room to spend all of his time in the light room, either because you're conditioning to like it or because you're conditioning him to dislike the darkness. And that's nice because it shows you that these circuits, that it's not just an individual neuron, but it's an entire neural circuit, and it's a behavioral output. You can make animals make different choices. You can take the device out, remove it entirely, and the animal will continue to prefer things it should not prefer, being in the light, because it's been conditioned. Its behavior has been changed fundamentally. And we can start to combine this with the autonomous neural control, the optogenetic stimulation, OGS, with the autonomous neural control, ANC, so that you can do this in a nerve using an optogenetic nerve cuff. And that's very nice because now instead of, so if you go in and you electrically stimulate a nerve, you're going to recruit the large fibers first and the small fibers last, because that's how electricity works, right? But if you go in there in the body, when it recruits a muscle or nerve fibers, it recruits small fibers first, large fibers last. When you do optogenetics, it does it in the same fashion. And so using optogenetics, we have a more physiological way of doing nerve fiber recruitment. And you can couple that with the autonomous neural control, and that becomes quite interesting in its own right. So those are two of the stimulating modalities that we're involved with. On the networking side, we try to do two things. One is we try to power these devices. So if we're going to make small implantable wireless devices, we need to figure out a way to deliver power to them, and batteries won't do, because batteries are large and cumbersome, and they run out of charge and need to be replaced. So we need to figure out a way of powering, and there's lots of different ways to do this, but what we do is we do far-field electromagnetic coupling. So you have ambient electromagnetic energy in the air. That's some amount of what you can get, but really you need more than that. So you can create a signal source, say a 1 watt, 2 gigahertz, 2.4 gigahertz transmitter. You can put it in your bedside table, and then whenever you lie down at night, your implant will pick up that 2.4 gigahertz 1 watt signal. And instead of demodulating it like you would with your Wi-Fi to extract the data for your internet connection, what it'll do is it'll just rectify that incoming signal and store it in a capacitor. And not just any capacitor, but a supercapacitor. More on that in a little bit. So there's ways of designing these circuits so that you can optimize the efficiency with which you capture power and with which you distribute power afterwards. And we spend a fair amount of time doing analog integrated circuit design for this purpose. The frequency selection is non-trivial, so we spend a fair amount of time looking at frequencies. When you think about an implantable device, you'd think, well, the lower the frequency, the better, because the lower the frequency, the less attenuation you're going to get through tissue. And that's true. But then the lower the frequency, the bigger the wavelength, and that means the bigger the antenna. I learned that from Dr. Bernhard in the back years and years ago. And the antennas get too big, it's kind of hard to fit them inside the eye, right? So you have this trade-off. If you have a fixed size that you have to work with your device, say a centimeter, well then a big wavelength is going to force you into an electrically small antenna, which is challenging, though perhaps not insurmountable. At the same time, a low wavelength is going to give you the advantage of the tissue attenuation. Add to that a third factor, which is that the rectifier for that power is on chip, and rectifiers work more efficiently at low frequencies than high frequencies. And you can basically start to add all these together to create a figure of merit that tells you where is the optimal frequency to operate in for a particular device of a particular size in a particular part of the body. And most of the time we go to 2.4 gigahertz. Lately we've been forcing some things at 900 megahertz, and it's been a little bit challenging. A few days ago we were talking about building a 60 gigahertz device. And so there's lots of different frequencies, and the optimal point occurs differently. It occurred to me that throwing up an antenna in front of my old advisor was always a precarious thing to do, but I will say that this is a meandering dipole, and that's all I will say about it. But we do a fair amount on antenna design, which we don't know how to do well, and one of the reasons why I was excited about coming here for this seminar is because it's an opportunity for you to bring Henry, who is a student in my lab in the back, who does antenna work, and introduce him to my old advisor and start to work together. So full circle. It's kind of nice. Anyway, so we do some antenna design work. These are efficiencies that we were looking at a year ago. More recently, Henry's been able to get minus 6 dBi efficiency on a 900 megahertz antenna of these dimensions, which he's very proud of. And he can show you. The way that we demonstrate this when we bring people into the lab is we take these antennas, we couple them with a rectifier, with a well-matched rectifier circuit, like the one shown here, and then we hook it up with an LED, we drive that LED. And you can do it for different sorts of devices. So from the very small, like a intraocular pressure sensor for glaucoma, there's the LED lighting up. A larger cardiovascular sensor, there's the LED lighting up. Now further away, a TMR sensor for EMG recording, further away still, and then if you can cheat with the antenna, make them very long and flexible under the skin, well, you can power them from 25 feet away in the lab with a 1 watt signal source. So it all depends on what sizes you can play with, what materials, what form factors. On the telemetry side, we began by looking at, if you have an implantable device and you need to transmit, let's say that you're recording data at 10 kilohertz, right? And so you're transmitting 10 kilohertz with, say, 10-bit resolution to keep the numbers simple. So you've got 100 kilobits per second that you're going to be transmitting. Your transmitter is going to use 99% of the power consumption of your device. That's just the way it goes. Transmitters use about a milliwatt. You can do everything else for tens of microwatts. And so the transmitter completely overwhelms your device power consumption. And that power is coming from that wireless coupling, which is not very efficient, right? So what do? Well, the first thing that we started to do is to say, well, why have the transmitter on all the time? If you can make a quick turn on and turn off transmitter, then have it turn on, transmitter data point, turn off, and then wait until a few hundred microseconds later when it's got the next sample. Or alternatively, if you don't need to be real time, have it accumulate data for a few seconds, turn on, transmit all in one burst, and then turn off again. So if you have multiple devices, you have some sort of time division multiplexing scheme in place where you can have these devices talking intermittently or spoken when spoken to from an external base station. So that's one implementation that we have. But another one, we had this idea that we're wirelessly powering these devices, right? So we have an incoming RF signal. And all the energy to transmit comes mostly from generating this onboard RF signal that you're going to transmit outwards. But we've already got an RF signal that's coming in. So why not take that incoming RF signal, put it in a flip-flop, divide it by 2, and retransmit it back out with a modulation? And so we started talking about this last year. We designed it on an integrated circuit. We fabricated the integrated circuit. It came back 3 weeks ago. We've tested it. It works. And so we now have gone from a transmitter with a 1 milliwatt power consumption to an equivalent transmitter with a 97 microwatt power consumption. So that's an order of magnitude reduction. It's a big deal. It means your battery will last 10 times longer or be 10 times smaller. And then we also designed the receivers, of course, that go on the outside and interface with the implanted devices. We did this all on an iPad because it was a nice interface that you could carry around to demos. But we've got various different versions working on it. The power storage is another area where we spend a lot of time. So the batteries have this problem that they're not rechargeable, or that if they are rechargeable, there's a finite number of times you can recharge them. And even then, charging a battery is tricky, especially inside the body. If you overcharge it or if you don't charge it at the right speed and so on, you can get the battery to do bad things, overheat, explode, and so on. So a capacitor would be much better, but capacitors don't have the energy capacity of batteries. So what we wanted to do was to come up with a better power storage design. And this is what we came up with. This is a photograph of a supercapacitor. We have a postdoc from China. His name is Chuizhou Meng, who came over. And this supercapacitor, you can see here, it's got 2 contact pads. It's 700 by 700 microns. It's got a thickness of 50 microns and a capacitance of 1 millifarad. Right? So that's a lot of capacitance in a very small volume. The catch, of course, there's 2 catches. One is that it's got a very high effective series resistance. So if you try to pull any kind of current out of it, you lose a lot of voltage just in your capacitor. The other catch is that the breakdown voltage is about 0.8 volts. So I talked about those circuits that we designed earlier for power management. A lot of what those circuits do is they boost the 0.8 volts or the 0.1 volt as it discharges. They boost it up to the 1.8 volts we need to run our integrated circuits off of. And at the same time, they take the charge on the supercapacitor and maintain that charge at the right voltage on a conventional capacitor. So when we have a high current spike, we can pull it from the conventional capacitor, which has a low effective series resistance, and not from the supercapacitor. So we can optimize efficiency by designing the circuits intelligently around that. So that's how we address the wireless component, or 2 of the ways in which we address the wireless component. The next piece is the miniaturization, and a lot of effort goes into this. The first approach, obviously, with miniaturization is by putting all of the intelligence of these devices on an integrated circuit. This is our most recent tape-out run. So are there any circuit designers here in the room? No circuit designers here in the room. Or are you just shy? All right, she's shy. All right, so okay, for the circuit designers in the room, you'll know what this is. For everybody else, when you get a chip inside your phone, when you watch, well, it's not Steve Jobs anymore, but when you watch whoever it is, the new guy, giving a talk for the new iPhone, and he talks about the new A7 chip or whatever, right? Those chips are custom integrated circuits. That's what we do on a much smaller scale. We're making custom integrated circuits for medical applications. And so when we come up with a design, we have multiple different projects, and we come up with lots of different chips. Each one of these little squares is a different chip for a different application. And this is our recent submission. This is where that new transmitter is on, and so on. And so, for example, we have a chip on here for glaucoma. It's 700 by 710 microns, and it allows you to record intraocular pressure when coupled with the pressure sensor I showed you earlier, and transmit that out of the body. We have a chip for seizure monitoring that allows you to record electrical activity in the brain and transmit that out, and transmit detected seizure events early in their onset, early enough that we might be able to intervene in the seizure before the seizure propagates. We have a chip that allows you to monitor pressure continuously in the heart, so that you have an early warning system for congestive heart failure patients of heart failure. We have a chip that allows you to record electrical activity in the muscle, so that you can have patients that have been amputated, for example, where you can reinnervate the muscle. More on that in a little bit. And you can record that muscle activity as a proxy for brain activity and use that to control a robotic limb. And all of this goes back to the original theme, which is that a single patient will not have a single pathology over the course of their life. An individual patient may at one point have glaucoma, and at a different point, which is the second cause of blindness in the world, they may also be epileptic. Right? And so you don't need to treat one in isolation of the other. You can create a device for one, and another one for the epilepsy, and another one for the congestive heart failure, and another one for the neuromuscular condition like Lou Gehrig's disease or multiple sclerosis, or for the prosthetic arm that they need if they're a warfighter coming back from Iraq or Afghanistan with an amputation. And all of these devices can be networked together into a network of wireless devices that treat the patient as a whole rather than as a collection of distinct and unrelated pathologies. So the vision here is that we partner up with NEEDS. NEEDS is an initiative at Purdue, nano-engineered electronic device simulation, which takes nano devices that are available, resonators, biosensors, a number of different devices, and models them using tools like SPICE in the same way that we monitor or model transistors, or resistors, or diodes, or other components which we put on integrated circuits. And that allows us to design a full biological implant not just by designing the chip and then separately designing the sensor and the actuators that I've shown you and then trying to glue them together in one package, but rather by designing all of that functionality into a single monolithic integrated circuit. Right? So you can design it all together, you can then fabricate it all together, and we've actually started to do this already. So we've got some devices where you take a transistor and you coat the gate with an analyte that responds to a particular biomarker, and that modulates the voltage at the gate the same way that you would ordinarily, except it does it with a concentration of a particular protein that you're interested in. And that's a reversible reaction, and so you can use that to track protein concentrations in the body. You also have a device that we just fabricated. It's a nanoresonator. And in the nanoresonator, you space doped channels at a particular distance. So you create an acoustic resonant chamber on the silicon, which you then pattern the metal over it to create a resonant chamber, and you can get high quality factor resonances. So the best resonator that you can make on a standard CMOS process is an inductor. It's got a quality factor of about 10. That's the ratio of the energy used to the energy lost. These have quality factors of over 1,000 measured quality factor. So it's a 2-order magnitude jump after 20 years of no jump at all, right? So it's a big deal. And these are nanostructures which we can now model through needs into our systems, fabricate, and integrate. And we can then package them, right? And I have this one here. If anybody wants to look, I've got this one actually here. Why don't I pass it around? So this is a device we made for muscle activity sensing. And it includes the microchip that we fabricated that I showed you earlier. It includes the supercapacitors that I showed you earlier. It's got 2 contact points and an antenna. And I use the word antenna loosely because it looks like an antenna, but it doesn't actually radiate. Ordinarily, I wouldn't say that, but present company, I felt like I should. Anyway, my student is blushing and contemplating leaving my lab. But anyway, so this is the device. The package is really cool. That's what I wanted to show you. We can take these and we can make them smaller, especially as we start to get rid of some of these off-chip components and bring them more and more and more on-chip. So we can make them really small. Here's one that's 2 millimeters, and the board itself is only about 800 microns long. The rest is a loop antenna, which sort of radiates. We can make them small enough to go inside of a mouse, and not just anywhere in a mouse. We can make them small enough to go inside the eye of a mouse. So this is really, really small. Right, this is the capacitive pressure sensor I showed you earlier, fabricated in collaboration with Bill Chappell at Purdue. There's the loop antenna, and that's a complete wirelessly powered, wirelessly transmitting 0.5 millimeter mercury resolution intraocular pressure sensor in a mouse eye. It's small, right? We can make them smaller. So here's a device. Here's a penny. There's the Lincoln Memorial on the back of a penny. And as you zoom into Lincoln, you'll see that next to his head, he's got a microchip. And off of that microchip, we've got a wire, which doubles as an antenna and an electrode. So you can record using it, or alternatively, you can coat the tip of it with a biomarker and use it for biomonitoring and telemetry. I tried to coin this one. I tried to call it an antectrode. I thought that was really cool. Somebody told me I needed to come up with a new word for this, and I came up with that word, and I used it at DARPA. I put it on posters. It didn't take. So anyway, but it does work. So you start to think about these sorts of size scales, right? Especially when you can incorporate all these different technologies on a single system. And now you start to see that using that magnetic insertion system I showed you earlier, we can inject these anywhere in the body, and we can inject them in a network fashion, right? So if we had the right sorts of capabilities, which we do to some extent in the partnerships that we have at Purdue, but we're growing, right, then you can envision that we'd be able to drive forwards this area of personalized medicine around distributed physical networks of wireless nanoscale sensors and actuators. And so we're hiring. And for all you future faculty, I'm not poaching existing faculty. If you are an existing faculty member, come see me. But for all the future faculty, right, so we're talking about nanobiosensors. We do a lot of modeling with nanobiosensor, but we don't do a lot of fabrication. So we're dramatically increasing the fabrication facilities that we have at Purdue. And if you know people who aren't in this room who might be interested, please feel free to spread the word and give them my contact information. A lot of what we do right now is electrical sensing and mechanical sensing. We're moving towards hydrogels, but those aren't very nano. But if we start getting into magnetic sensing, and particularly enzymatic-based sensing, like what I was showing you with the coating of the gates of the transistors, then I think we can start to do some really interesting things in terms of this nanobiosensors, packaging, Everything that the FDA, from a regulatory standpoint, cares about is hermeticity. They want to make sure that the device and what's inside it is not going to hurt the body, which is fun because the devices almost never hurt the body. It's actually the body that hurts the device, right? You take electronics and you put it in a warm saltwater solution for the next 80 years. That's not good for it, right? It's the worst possible thing you can do to an electronic board. So we worry about the hermeticity because we want to protect our electronics, not the patient. And so we talk about ceramic packages and titanium packages and all sorts of different materials, but these packages aren't very interesting from a functionality standpoint, in the sense that it's very difficult to incorporate the sorts of microelectromechanical system structures that we want to within those packages, that they're rigid, not flexible. So we want to have these features, and that means we need to move in the polymer direction, and polymers are by definition almost not hermetic, but they're near hermetic. So trying to make these package materials in ways that are interesting, with incorporating technologies that are exciting in ways that are hermetic enough for the amount of time that we need, this is a major direction thrust as we move forwards. And energy harvesting is the last one I wanted to talk about. So we do a lot of inductive coupling. This is the only one that's approved by the FDA right now. I talked to you a little bit about RF powering. You can do it with high-intensity focused ultrasound or optical, but these really don't have a lot of patient compliance. And what I mean by patient compliance is that they require the patient to do something. So if a patient has to take an external charger and hold it up to their implanted device to charge it, you're almost guaranteed it's not going to happen. It's like the glaucoma story where the patients get drops. The drops will take care of the glaucoma, and yet the patients almost always go blind. It's because they don't take their drops. And when you ask them, they lie to you and tell you that they did. Patient compliance is the bane of the medical world. What you need is a device that is patient-proof, right? So you need to get away from devices that require you to do something and towards a device like RF powering, which is more or less somewhat patient location independent, that you might be able to put it in your bedside table, and as long as you get into bed every night, it'll charge your device. And preferably even towards a device which harnesses the thermal and mechanical energies that exist already in the patient and uses those to power the implant. So we have 3 new faculty slots in these 3 new areas, and with them, the sense is that we can really begin to enable these wireless networks. So here's an example. This is Claudia. She's a patient up at the Rehabilitation Institute of Chicago. This is through the partnership with the DARPA grant that I'm the PI on. And she has a series of sensors located on her left breast. So when she lost her left arm, not in combat, motorcycle accident, when she lost her left arm, she, it transformed her life, as you can imagine. But what the doctors noticed was that the nerve that still went down the arm was still there. And the pectoral and lateral muscles, they're still there. They're just useless because there's no arm to move around. So why not take the nerve that goes to those muscles and remove it, de-innervate the muscle, take the nerve that used to go down the arm, connect it to the muscle. Over time, it'll regrow into the muscle and re-innervate the muscle. And then, when you tell Claudia to open her hand or extend her arm, these muscles contract in unusual ways. We can use the device that's getting passed around, use 10, 12, 16 of them, in the muscle, positioned throughout the muscle body, to extract the electromyography, the muscle electrical activity, that we can then transmit wirelessly to a prosthetic arm, allowing her to control a robotic arm with more degrees of freedom and more sensitivity, more accuracy than anything that's ever been done out there. And this is not, I can say things like better than anything ever because it's not my work, it's theirs. It's the rehabilitation's work. What we're doing is we're looking at that device and saying no patient is ever going to use that because those electrodes, right, first of all, they're positioned on the skin. When the muscle contracts, it moves with respect to the skin, so your signal source is moving with respect to your amplifier, and that does bad things to your algorithms. Second of all, every time you go to bed, you take the electrodes off, you put them back on in the morning. They're in different locations. You've got to reprogram the whole system. It's a big, cumbersome pain in the neck, and that means patient compliance goes to zero. So you actually take one of these arms. The first one cost $100 million to develop. Only DARPA would spend $100 million that way. And it was really cool, but it was just the first one, right? And it was buggy. DECA made a better one. That's the one you just saw. You take these arms, you give it to the patient. They think, wow, this is really cool. They show all their friends. They go home. They go to bed. They wake up in the morning. They throw it in the closet. They grab their hook, and they put their hook on because the hook is lightweight, and it works. You don't have to do anything fancy. Now, if we use these electrode arrays, we can get these signals reliably, repeatedly, so all she's got to do is just put the arm on, just like a hook, and it'll automatically work because it's already been calibrated, and that only has to happen once. So those are the sorts of things, right? Especially when you start to couple them with other devices and other conditions that we're talking about here. So this is not just me, of course. It's mostly not me. It's the students, it's the partnerships, the collaborations, the sponsors. They're all important sponsors. Primarily for the work I've talked about today is DARPA, Cyberonics, and Howard Hughes Medical Institute, and a number of graduate students, postdocs, research scientists, technical staff. They do all the real work. But thank you very much. Yeah.
How strong of a magnetic field do you use to pull the electrodes down?
It's a good question. I don't know. So what we did is, so you know what a railgun is?
Yeah.
Okay, so basically what we did is we built a railgun. Initially what we wanted to do is try to figure out whether or not we could even get these things to move. So we made the electrodes a little bigger, 100 microns across, because 20 microns was a little challenging. There's not a lot of material there. And then we took a coil we just wound and wound and wound and wound. And then we got a capacitor, we charged it up to 400 volts, and we shorted it across that coil. Blew sparks and all that good stuff, and shorted out between the turns and everything else. But the electrode came out and embedded itself in a door on the other end of the lab. So that was fun, right? And so it's not often you get to actually build guns in the lab and call it medicine. But anyway, so we did that. Then somebody wanted to see if we could do a craniotomy. And so we took an animal. It was euthanized. It was already dead. But we just took the electrode, and we were actually able to penetrate through the skin, through the bone, and into the brain, and bury in the base of the skull. So we had too much juice. And so what we did then is we calibrated a curve for different electrode thicknesses, different voltage amounts, and different current or numbers of turns and coil diameters and so on. And so we had sort of a tuning curve for what depth you wanted to get. You'd apply this much juice versus a different amount of juice. The next step was to try to figure out if this was all really being done as efficiently as possible. So I have a new PhD student that just started a couple months ago, and he's actually started going back to Maxwell's equations and trying to figure out, all right, how does this actually all work out? And it's actually a lot of fun, because it's been a while since I've gone through these equations. Sorry. But you know, I see this, and so he looks at this and he says, well, actually, you know, the electrode initially is at a stop, and so you don't want to apply the maximal force there, because a lot of it's going to go wasted. You want to accelerate it slowly, and you want to have increasing field as the electrode moves. So you stack the coils, so there's few turns up here and high turns up here, and he comes up with these pyramidal structures that actually work really well. The next problem that we have, so we don't know what the magnitude of the magnetic field is. It's mostly trial and error, but now we're just starting to try to figure out what the theory behind it is. What's interesting is that he came up with an idea for having a ferromagnetic holder with a DC magnet at the tip that holds a non-magnetic electrode. And so you can put this in the same barrel and fire it, and then just at the critical moment, you have a DC magnet around the outside, and it opens the holder. And so the non-magnetic electrode will proceed. It's more of a javelin throw than a magnetic field pulling it in, but it'll proceed into the tissue, and what you're left with is a non-magnetic electrode, which potentially you could then use to measure single unit electrical activity coupled with fMRI activity in an MRI environment. Yeah, sure.
When you were talking about how you were powering the devices, you talked about how one of the frequencies you used was with 2.4 gigahertz.
Mm-hmm.
Now that's a pretty common frequency, right? It's used in cordless phones, et cetera. And if you're also trying to transmit out the data on there, are you worried that, or
Yes.
is it possible that, you know, people moving about, entering areas with those fields is going to interfere with the monitoring of?
So if you're trying to make a device that's going to see clinical adoption, right, and the clinical impact is what we're all about, then you need to operate in a frequency band that the FCC is going to let you to stay in. And those are ISM bands, industrial scientific medical bands. The reason there's a lot of stuff at 2.4 gigahertz is because it's an ISM band, which means you can do just about anything there. And so you've got Bluetooth, and you've got Wi-Fi, and you've got us. So what we've done lots of different variations. One time, the chip that we did a year ago, we had an OOK transmitter, which is on-off keying. So the transmitter's on when it's a digital 1, and it's off when it's a digital 0. And it was at 2.4 gigahertz. And we thought, oh, great, you know, on-off means that it's off half the time on average, and that means that you're saving half the power. And that's kind of clever, and it's not a very complicated modulation scheme. It's easy to modulate. It's perfect, right? Why would you do anything else? So we fabricated it, and the damn thing didn't work. And the reason it didn't work is because anytime the receiver sees a 2.4 gigahertz pulse, it registers a 1. But if you're in a room in which somebody's cell phone is downloading data and you got somebody's Bluetooth headphones going off all the time, every time each of those devices sends out a spike, the receiver picks it up as a 1. And so we had huge error rates, which drove our data transmission rates down because we had to retransmit everything all the time. So you do have to worry about it. More recently, we've done this divide by 2 scheme, which is nice because you can power it at, say, 2.4 or 900 megahertz, and then you retransmit at half the frequency, 450, which is out of band. But that's okay because these medical devices are so low power in their transmission that they're actually below. So the FCC has guidelines for intentional radiators and unintentional radiators. You're transmitting below the intentional radiator, you can get away with it, which is one of the things that we're doing. You can also shift things a little bit, so you can try to be in the mixed band on the transmit, which is 430 megahertz, and a 900 megahertz on the receive. On the power. There's lots of games that you can play, but yes, it is generally a bad idea to receive and transmit at the same frequency, but you can do it. The intraocular pressure sensor that I showed you does just that. That's an FSK transmitter, though, so that's always on. It's easier to pick out the noise. Yeah.
What's the power consumption of the LED light up, and what's the transmission power of the transmitter?
So the LED will light up with as little as a few microwatts. It varies, though, of course, right? I mean, you saw the LEDs go super bright and they get dimmer. So you can put milliwatts in it. I mean, they're rated at milliwatts, but you can light them up with just a few microwatts. Generally speaking, what we need for our medical devices is, so we're looking at a peak power. It used to be a milliwatt. And so we were thinking if it was a milliwatt, we could have it on 1/10 of 1% of the time. So 0.1% of the time, because the rest of the time it's off between samples. If you can get the ramp up and ramp down, which we haven't been able to, then you could get the power consumption down to an average power consumption of a microwatt. The average power consumption is what you actually care about. With a new transmitter, we've got our quiescent, or our peak power consumption, down by a factor of 10. So that helps quite a bit. And it means that we only have to be on 1% of the time instead of having to be on 1/10 of 1% of the time. And now I think it's more realistic that we'll get an average power consumption of a microwatt. But that's the goal, is 1 microwatt average power consumption, which is a little less than what we need to light up those LEDs.
I mean, what's the peak power of the transmitter?
The peak power of the transmitter is 96 microwatts.
Microwatts.
Mm-hmm.
Why does my glaucoma device need to talk to my epilepsy device and my heart failure device?
Because let's say that you have a glaucoma and you have a cardiac condition. Pressure in the body is linked together. So variations in pressure in one area are linked to variations in pressure in the other, and reducing pressure in one area can track pressure in another or increase it in another. So they're linked. Drugs that you take to affect your blood pressure might increase the pressure inside of your eye. So these diseases can be linked, particularly if you start to think about multiple different conditions inside of the brain. So if you have a traumatic brain injury, or you have an addiction, but you also have epilepsy, you're talking about neural recording and stimulation for multiple regions in the brain. There's no reason to have one device that does multiple regions for one condition and another device that does multiple regions for a different condition. Why not have a network of sensing electrodes and a network of stimulating electrodes that can address multiple different pathologies now? And as you age and you develop Alzheimer's, we can start looking at doing things there as well. So it's a way of centralizing all of these different things. I have a friend of mine with whom I work with on the glaucoma who had high blood pressure. So he started taking medication for his high blood pressure, and it's irreparably damaged his neuromuscular junctions, so that he now spends 3 hours every day stretching out his muscles because he wakes up in a cramped fetal position every morning. And that's a side effect of the cardiac medication. So the downside of treating these pathologies individually is that patients with one end up exacerbating the others because of the side effects of the drugs they're taking for that one condition. The upside of treating the patient as a whole and networking these devices is that we can try to get around those sorts of problems, which are surprisingly common. It's a good question.
So how do you see that changing the entire field of medicine? Because it's going to have an impact on the specialists individually. Mm-hmm. So how do you see that interaction between them changing?
So let's take a condition like epilepsy, with which we spend a fair amount of time. Even leaving aside the networking properties, epilepsy affects 1% of the population. Of that 1%, that's 3 million Americans. It's 1% worldwide, but just in the United States, it's 3 million Americans, roughly. 30 to 40% don't respond to drugs. 30 to 40% respond to drugs, but the side effects are so severe that they end up not taking their drugs. The end result is you get about 54,000 deaths from epilepsy every year in the United States. That's more than breast cancer. That's because drugs don't work. They work for some people, but for a huge percentage, about 2 million Americans, they don't work, or they don't work well enough to make it worthwhile. These same people who stop taking the drugs because it's not worth it living with the side effects to stop the seizures can often die from a condition called SUDEP, sudden unexplained death from epilepsy, where you go into a persistent seizure and your cardiac arrest, respiratory arrest follows. And it's an unexplained, that's why the unexplained death, it's kind of terrifying, right, for a medical acronym. Kills thousands of people every year. So the current treatments are not sufficient. Folks who go on drug treatments, whether it's for pain management or for epilepsy or for Parkinson's or whatever, over time the body becomes desensitized to the drug. Not to mention that the drug is distributed throughout the body and you only need it one place. These approaches allow you to target therapies so that you're going right where you want to be going. You want to deliver the therapy only when you need to, and you avoid chemical desensitization because there is no chemical to become desensitized from. So the notion that you can move away from pharmaceuticals and that you can move towards a non-drug, a drug-free therapy program, and that you can do this in a way that you're targeting it to the area that needs it. These are all revolutionary ideas. And that you can network it together so that you're looking at the side effects of a particular treatment in other regions, and you're treating them all as a whole. All of these are different ways of looking at medical practice than what is done right now. And we have quite a bit of support. We have partnerships with a group called Goodman Campbell Brain and Spine. It's a merger of all the neurosurgical groups in Indianapolis, which is now the largest neurosurgical group in the United States, and one of the best. And we partner with them to do human clinical trials, to try these things out on humans. And our sense from conversations with the doctors or with industry, is that there's a willingness to begin to explore and commercialize and deploy these treatment modalities that didn't exist maybe 10 years ago. That's partly because the technologies are much further than they were then, and partly because a sense of the limitations of more and more and more money at high throughput drug screening has not yielded increasingly better results.
Okay, so let's thank Professor Irazoki for a very interesting